This procedure begins with growing Drosophila third instar maggots with the appropriate genotype on food containing all trans retinol. Each maggot is pinned out head and tail, then flayed flat. The brain and posterior nerves are then cut to stop rhythmic motor activity. Next, the blue LED light source with control box is adjusted and focused on the preparation. Finally, use a sharp electrode to impale muscle 6, then deliver light pulses with a voltage source.
Hi, I'm Nicholas Hornstein from the lab of Dr. Leslie Griffith, the Department of Biology at Brandeis University.
Hi, I'm Stefan Pulver, also from the Griffith lab.
Today we will show you a procedure for evoking synaptic potentials with Channelrhodopsin2 at the neuromuscular junction of Drosophila larvae.
We use this procedure in our lab to study synaptic physiology in Drosophila. We also use it to teach students synaptic physiology in teaching labs.
So, let's get started.
To get started with this protocol, maintain UAS Channelrhodopsin2 and OK371 Gal4 fly lines in separate bottles containing standard fly media. Collect virgins from the OK371 Gal4 fly lines and males from the UAS Channelrhodopsin2 lines. Next, make fly media that is mixed with 1 millimolar all-trans retinol, or ATR. To do this, melt regular fly media in a microwave for approximately 1 minute. Once melted, allow the media to cool for about 30 to 60 seconds, and then add 100 microliters